couple of years ago, it was quite popular in the press to harass the ID proponents with the idea of junk DNA. That is, if you're making a teleological argument that God exists through information or intelligent design, we'll give you examples of dysteleology. We'll give you
Yeah.
examples of poor design. And for example, they say, what about junk DNA? It serves no purpose. How do you explain why God would put junk in there? And so, first, what is junk DNA, and does that term, is it even still used anymore?
Oh boy, this could be a whole show on nothing but junk DNA, because already there's like 5 or 6 different ways to approach this, and I'm not exactly sure where I could start.
Yeah.
Let's say it this way: junk DNA came about from theoretical work in the 1950s.
Mm-hmm.
JBS Haldane is looking at natural selection and mathematics, and he says you can't get millions of changes in millions of years. We got a problem. So 10 years or so later, Ono and Kimura, these Japanese scientists, they came up with this idea that there's junk DNA. That is, okay, look at that. Only 2% of the genome codes for protein. The rest of it is junk. Therefore, it's not subject to natural selection. Therefore, it can mutate randomly as much as you want, and we don't have to worry about mathematics anymore, because 98% of the changes don't matter. And that held on until we sequenced the human genome in the 2000s. And we realized soon after that that most of the genome actually does something. It might not code for protein, but most of it codes for RNA. And the RNA is sort of like, if we were a computer, our ROM would be the DNA, the read-only memory, which actually isn't true because it can change. But let's just say it doesn't change, it's a read-only memory. The output is the proteins. But in between is the random access memory, the RAM. The RAM is the RNA. It's at the level of RNA where all the decisions are being made. The RNA is controlling which genes are made into proteins, et cetera, et cetera, et cetera. It's all this massive computer decision-making network, which is at the level of RNA, and most of that is RNA that doesn't turn into protein. So most of the DNA is actually made into RNA. In fact, most DNA is made into several different pieces of RNA that their code overlaps, but these things have nothing to do with one another. They're completely different parts of the genome. So if you mutate that letter, you've just mutated 2 or 3 or 4 different messages that do different things. So one mutation might have something to do with how well you see in the dark, or whether or not garlic gives you the hiccups, how tall you are, and what your hair color is. Now, I totally made all that up, but
Yeah.
Each letter is involved in more than one process, meaning that mutations change a lot of things that no one expected. So, but the thing is also, I have no problem with junk DNA for 2 reasons. One, things have broken over time. Okay. But 2, part of the information content of the genome is not for making proteins, it's for holding DNA in a certain configuration. So the genes that work together are near each other in 3D space. That's an efficient, optimal design. But the things that link them, the sequence isn't that important. Maybe you need a bunch of G's and C's in one area to create a bend. Okay, fine. Well, whether it's a G or a C, it doesn't really matter. So all that can mutate like crazy. It's not junk because it's structural, but the exact sequence isn't that important. The general sequence is important, but not the exact sequence. But even within a gene, if you change one letter in DNA that's in an important protein, it probably won't affect the protein sequence at all. If it does affect the protein sequence, it's just one amino acid out of thousands of amino acids. It probably doesn't have any effect on the enzyme. So there's plenty of room for change, plenty of room for flexibility, if I can use that word, instead of junk.
Okay, so we're going to throw the word junk out then.
It should be thrown out, yeah.
We'll call it support DNA.
Support DNA, I like that.
RNA, you mentioned that a few times as being created from DNA.
Yes.
Is there more than one kind? How does that work?
Oh, there are lots and lots of kinds of RNA. The one we're most familiar with is called messenger RNA.
Mm-hmm.
If you take a piece of DNA and just transcribe it into RNA, um, that RNA is run through a ribosome, and 3 letters at a time becomes 1 amino acid in the protein.
Mm-hmm.
In bacteria,
Yeah.
in humans, it's a whole lot more complicated because the RNA has spacers in it. They're called introns, have to be cut out, and then what's called the exons have to be joined together to make your thing that's going to be turned into a protein.
So it has to
But
be.
we now understand that these different little sections are used in different proteins. And that's one of the big mysteries. When the human genome was first completed, all the geneticists in the world, actually, Edwin Burney, he had a betting book. And he was taking bets from all the top geneticists in the world on how many genes they thought were in the human genome. Not a single person got it right.
Wow.
The closest was a graduate student, and she guessed about 28,000. Everyone else is up in hundreds of thousands range because the human body makes like 300,000 different proteins. Turns out we only have about 22,000 genes.
So that you had to recombine them in different ways to.
The recombining of all the parts
Mm-hmm.
leads to all these different proteins, meaning that the intervening sequences, there are codes in there that say use this part of this protein, or let's see, use this part of this gene in this protein in this cell type at this stage in development under these conditions. So all that junk is actually coding, and there's computer codes in there. And our very few number of genes can produce a fantastically huge number of proteins because of the way the computer code runs.
Do you see evidence for design in this system?
Oh, it's all design, all the way through. Article on creation.com called Splicing and Dicing the Human Genome. If you
Mm.
just go on creation.com, type in splicing, I'm pretty sure that'll be the top article. And it explains the hypercomplexity of the genome and how it defies all naturalistic expectations.
Well,
See,
not
if
surprised.
life were simple,
Yeah.
evolution might be possible. That's true. But the more complex life is, the less possible evolution is. And today, all we understand is life is hyper complex.
Mm. To the level we don't even understand it yet.
No.
Yeah.
In fact, I have my favorite talk I've ever given is called the high tech cell.
Mm.
It's available on creation.com also. But what I did was I compared the human genome to a computer, and very quickly we got to a point where the computer is more complicated than any computers humans have ever made.
Mm.
And I made a prediction. I said, I think that our understanding of genetics is going to affect computers in the future because we're good copycats and we see something, oh, that's really smart. Let's do it that way. And probably our understanding of the information content of the cell is going to affect the way we design computer chips and the way we write computer programs.
Yeah, I know that several years ago there was a computer scientist in Israel who was talking about making genetic computing, and
Oh
he
yeah,
talked
and we're
about
doing it.
potentials. Yeah,
Yeah.
yeah.
But even on a silicon level, the way the information is processed in the cell is going to affect our computer systems. See, DNA computing is really cool, but it's going to be a little slow.
Mm.
You can store a lot more information in a test tube than you can in a similar size computer chip. That's true. But accessing the information, it would take a while to find it, to extract it, to read it.
Mm.
But DNA storage technology is really cool too. Put a bunch of messages on DNA and freeze them and bury them in Antarctica, and that information is going to stay around for a very long time.
I once read that DNA is trillions of times more efficient than any computer chip ever invented for storing information.
Yeah.
And the same scientist said that a teaspoonful of just DNA would hold the information for every species that ever lived and every book ever written.
Plus the internet.
Yeah, I actually
Yeah.
recently entered the internet last week, so I just want to let you know that I have been using my time fruitfully during this lockdown. Yeah. You mentioned earlier about the mitochondrial DNA. in the origin of that. We talk. You read sometimes about a Y chromosome atom. What does that mean, and is there evidence for that?
Let's start with the mitochondria first.
Okay.
Back in the late 80s, some scientists were looking at this little piece of DNA that you only get from your mother. It's called the mitochondria.
Mm-hmm.
The mitochondria are the things in our cells that make energy from sugars. You have about 1,000 of them in each cell, and they have a little piece of DNA. It's only 16,000 letters long, so it's really easy to sequence. And because there's so many copies of it, we can get a lot of it quickly. So that was one of the first pieces of DNA people started sequencing. And they looked around the world at different people and they said, wow, look at that. We got all these people in Africa, and then as a subset in our evolutionary tree, all the people out of Africa. Oh, therefore, we must have originated in Africa.
Mm.
Oh, well, that depends on the mutation rate.
Mm.
And it depends on mutation rates being the same in all people at all times. And a lot of other assumptions went into that. But what they realized was there's only one female who's the ancestress of all people on Earth today.
Wow.
And that was not expected. Because if we came from a population that's split into humans and chimpanzees, well, that population would have had a diversity of mitochondrial lines in there. And it's possible that this mitochondrial lineage is found in chimpanzees and humans. And this mitochondrial lineage is found in chimpanzees and humans, which means that some humans could be more closely related to a chimpanzee than to other humans. But that's not true. It's completely the opposite.
Right.
Not only are all humans related to themselves, they're very, very different than the chimpanzees on the mitochondria. It would take only a few thousand years to account for all the differences we see within people. So mitochondrial Eve, the evolutionist, was recent. Decade or so later, they came up with the concept of Y chromosome atom, which is a biblical mistake. It should be Y chromosome Noah, but okay, they don't know their Bible. We'll give them a pass. But Y chromosome atom was the male ancestor of all people in the world, because see, the same thing is true for the Y chromosome. All men in the world are very closely related, and the human Y chromosome is extremely different. In fact, it's the most different part of the genome to the chimpanzee. It's radically different. And so, well, Y chromosome atom must have been recent. And how recent is the question? Well, just a couple of years ago, Ancestry.com, they got sent a test kit. And when they did their sequencing on it, they said, whoa, this is a person we've never seen before. And one person who happened to be African American, they traced their lineage back to a man named Albert Perry, who was a slave in South Carolina in the early 1800s. And Albert Perry had a Y chromosome that was different than anyone ever found. And so what happened was they had to add a new branch of the Y chromosome family tree. They literally doubled the amount of time to Y chromosome atom in one day.
Wow.
And all of a sudden that put Y chromosome atom and mitochondrial Eve in the same time frame, the evolutionary time frame. So for all these years, we've been saying, oh, Adam and Eve, they never even knew each other. Ha, ha, ha. They lived 100,000 years apart. Ha, ha. Well, they don't believe that anymore. Now, I don't think that their dating is right, and I don't think their position is right. But it's really funny how the date shifted radically with the addition of one more sample.
Mm-hmm.
So there you have it. The Bible says there's only one male ancestor of all people, and so does genetics. The Bible says there's only one female ancestor of all humans, and so does genetics. Evolution did not make those predictions.
Mm-hmm.
It was a surprise to everybody.
So why do you call him Noah instead of Adam?
Because during the flood, all male lineages went extinct except for Noah's.
I see.
And Shem, Ham, and Japheth, being sons of Noah, they inherited his Y chromosome. So we will never know what Adam's Y chromosome looked like, because all the people in the world today come from Noah.
Right.
And we can go back to him. Now, we might be able to go back to Eve's mitochondria, but it depends on how closely related those three daughters-in-law of Shem, Ham, the daughters-in-law of Noah, that's the wives of Shem, Ham, and Japheth. If they were all sisters, then all we know is their mother's mitochondria. If they're all like daughters of Noah, like if Shem, Ham, Japheth married their sisters, which, biblically that's not impossible,
Right.
then we only go back to the flood on the mitochondria. But if they were very distantly related, we could have up to 3 different mitochondrial lineages, and we could use that as a window to estimate what Eve might have looked like.
Mm.
But we can't necessarily know. But there is
Okay.
only one female, and that's what we need to know.
Uh, I would agree with that. The evolutionary geneticists have said, and this is a quote, Darwin's core insight was that organisms with disadvantageous traits would slowly be weeded out through negative or purifying selection, while those with advantageous features would reproduce more often and pass those features on to the next generation, positive selection. Do you see evidence for this evolution of DNA?
I see evidence for positive and negative selection.
Mm.
In fact, that's perfectly acceptable in the creationist model.
Mm.
Some organisms have more baby than others. Okay. Why is that a problem? Why is that difficult at all? I just wrote an article called Natural Selection in Paradise, where I claim that if the fall had never happened, natural selection would still have been in place in the world since creation. Because some organisms will have more babies than others, and it depends upon their genotype and their phenotype. It's not a problem. What Darwin did was he waited a long time in his book,
Mm-hmm.
and he finally said, I see no limit to the amount of change. Oh, wait a minute, Darwin, I can see purple elephants flying through the room.
Mm-hmm.
I can imagine anything I want, but imagination is not science, it's philosophy. This is why I said earlier, Darwin was a philosopher, maybe not a very good philosopher, but he wasn't a scientist. He was applying, I think, to the scientific process.
Mm.
And again, once you have living organisms, you can have natural selection, you can have change, you can add adaptation, but it doesn't explain where those things came from. So everything Darwin wrote in The Origin of Species, most of that is non-controversial. It's not a problem.
Mm.
The problem is when you try to extrapolate to the beginning of everything. In fact, he wrote, to speculate about the origin of life, you might as well speculate about the origin of matter. See, he was trying to avoid the origin of life question. But it's
Mm.
funny because today the origin of matter happens in the Big Bang.
Yeah.
So modern scientists certainly are speculating about the origin of the universe and the origin of life.
Yeah.
And yet everything in operational science tells us that these things aren't going to happen by themselves. So Darwin was actually careful. He knew the limits of his idea. He pushed it as far as he dared, and then he stopped there because he couldn't go any further. And yet today people go as far as they want.
So you're saying that there's aspects of it that you can agree with, but it's more the philosophy behind the theory that's the biggest problem with it. That the philosophy that
Yeah,
guides the theory.
yeah. Major aspects of Darwinian evolution I am completely in agreement with.
Okay.
It's the extrapolation to infinity, the philosophical application, the assumption of naturalism that I completely disagree with. And now, not just scripturally, but also scientifically, naturalism doesn't explain origins. And that's the title of his book, Origins of Species.
Mm-hmm.
And he's just wrong about it.
There was a controversial book that came out in 1972 by Niles Eldridge and Stephen Jay Gould,
Oh, yeah.
in which they formulated their theory of punctuated equilibria, saltationism. That is, that they said it was based on the admission that new species appear suddenly in the fossil record. They remain basically unchanged, then disappear. Is this a way for mainstream evolutionists to admit that the fossil record does not support the neo-Darwinian paradigm of gradualist evolution?
I think after Gould died, the idea withered on the vine.
Mm.
I don't know, I could be wrong, but I don't know any major evolutionists today that's talking about it. I think they ran away from it quickly. And their answer is, oh, yeah, well, the fossil record is just spotty. It's incomplete. It only preserves some things. That's why it looks discontinuous. That's why it looks saltational. Saltational means leaping.
Hmm.
Um, it looks like there's giant changes in species technology, but it's only because it's so incomplete.
And so,
And
yeah, so their hopeful monster type terminology was kind of like Fred Hoyle calling the origin of the universe the Big Bang.
yeah,
He meant
it's
it
a
as a
joke.
pejorative, but it turns out to be pretty accurate.
Yeah.
Yeah.
Yeah. So because there's not really any good genetic mechanism for creating vast changes or dramatic changes in
Mm,
one generation, people have just dropped it. Now the Evo-Devo people, that's the evolutionary development, the Evo-Devo people, they're looking at these genes called Hox genes.
mm,
And Hox genes they set up major patterns in the developing embryo. So the reason that I have a left and a right, and the reason that I have lungs and a heart and a liver is because at one point in time, a gene turned on and said, okay, everyone around me gather together. Okay, listen, everybody, we're going to become a liver.
mhm.
Or they say, you know, this is an eyeball, this is a throat, this is your gut, this is your legs. And they trigger and create downstream effects on all these different organs. Where you think about it, my left hand and my right hand, they're mirror images, but the cells in my hands have not been connected for a very long time. And something said, I'm going to make things that look the same. And what the reason that they're the same is because there's a clock. Something in my, as I was developing, something was timing events. 3, 4, okay, everybody, go. Start developing fingers. Dun, dun, dun. Okay, go. Now you're going to extend and have arms.
Mm.
And it's these very complicated clocks and timing events in the cell and in the embryo. Well, the Evo-Devo people said, okay, change one of those, and all of a sudden we can grow wings in place of arms.
Except in order to have wings, you have to have a change in the bone structure, the muscle structure, the respiratory system, the
Exactly.
metabolism. Everything has to change. That's not a one switch type deal, which would what Darwinian evolution would require.
What we've seen is that changes to those core computer modules inevitably create catastrophic effects downstream.
Hmm.
The later in development you make a change, the more survivable that change is. The earlier in development you make the change, the more likely it's going to just kill everything. So you can change the color of your hair.
If I
You
had
can change your height. You can change your skin color. Fine. You can't change how many arms you have,
Hmm.
or whether or not you have a digestive system,
Hmm.
or any of those major defining characteristics. That's where it falls apart. And so Evo-Devo was a flash in the pan. Everyone thought it was the bee's knees back, 20 years ago, but really they've gotten nowhere. Their biggest one that they have is, I think the biggest example I've ever seen is Antennapedia, where they've got a fruit fly who grows legs in place of its antenna.
Yeah.
Yeah, well, it's a non-functional fruit fly, and that's going to be quickly weeded out in the world. So,
So would you? We were talking originally about punctuated equilibria and the fossil record. Would you say that the fossil record better supports creationism than Darwinian evolution?
yeah, I would say so. Okay, and I say it this way: Darwinian evolution requires millions of experiments over time to produce new features.
Hmm.
I don't think that's debatable. But when we look at the fossil record, the greatest leaps in evolutionary technology are covered by the fewest steps. So look at the Cambrian explosion. You have all the major phyla of life, boom, they appear. Those are the greatest differences in living organisms, and they suddenly appear in the fossil record with hardly a trace of any ancestry. But how many evolution would have taken to make echinoderms and bony fish and cartilaginous fish and plants and things with backbones and jellyfish and on and on and on and on and on. Billions and billions, maybe trillions of evolutionary experiments would have been required to make these diverse life forms, and yet the evidence for that is absent. We do see changes within groups. Sure, fine. Like my specialty was in corals. There's one particular genera of coral in the Pacific and Atlantic Ocean. It's called Acropora. There's hundreds of species. And when you look in the fossil record, there's species we don't recognize today, but they kind of look like this one and kind of look like this one, which means that within this genus, or this family, I should say, this family, not genus, family of corals, we see all this morphing and changing over time in the fossil record. And today we have a bunch of species also. But they've all changed. They're all recognizable. You could literally walk up to a rock from the Cretaceous and say, oh, I know what family of coral this is.
Mm.
So changes within that family, fine, but no changes from one to the other. No evolutionary links between the families. They all just appear.
Mm. So you're saying that the Cambrian explosion is not a friend of Darwin?
No, it's a massive problem.
Yeah.
Massive. And the old thing, oh, well, that's because the ancestors were soft-bodied, and soft-bodied organisms aren't preserved in the fossil record. In fact, Darwin wrote in The Origin of Species, no organism wholly soft can be preserved, because he thought the fossil record accumulated slowly over millions of years. Therefore, anything soft would decay before preservation could happen. But we've got fossils of jellyfish and fossils of worms and fossils of all sorts of soft-bodied things. Darwin was totally wrong. And the evolutionists who try to say, oh, they were soft-bodied, they weren't preserved. Well, we fossils of soft-bodied things. So where's the evidence of these massive changes that you need? It's not there. In fact, if you look at the timing of the Cambrian explosion, we have a video in Evolution's Achilles Heels. You can also find it on creation.com if you go to our media site. It's looking at, let's take all the billions of years of Earth's history and put them on a football field. And we said, okay, the Cambrian explosion happens about the 90-yard line, because you have billions of years of just bacteria before that. And all of this explosion happens in less than the width of one stripe on the field.
Yeah.
Less than 3 inches of a football field. Boom. It's the same amount of time, or actually it's less amount of time than it supposedly took whales to evolve from land animals.
It's pretty impressive.
So yeah, mammal to a mammal.
Yeah.
In that same amount of time, or similar amount of time, you've got all the phyla of life evolving from nothing. And I'm sorry, that's a problem for them, not for me.
So you're saying that the tree of life is less like a oak tree and more like a fir tree?
Actually, the tree of life is more like a forest.
Uh,
It's not a lawn. It's not a creationist lawn where each species is created independently.
mm-hmm.
It's a forest of trees. Each tree is created independently, but each tree has a lot of branches on it.
I see.
So dogs and wolves and coyotes and African jackals, that's all. They can all interbreed. That's one species or one kind that's divided into separate species today, but they're still reproductively compatible.
Mm. Okay, well, getting back more into the science side for my students,
Okay.
you talked about changes in proteins. Is it easy to get a protein to fold into a usable shape?
Take 2 amino acids and stick them together.
Mm-hmm.
They can twist around like this.
Right.
Take 3 amino acids and stick them together. You have 2 twisting areas now. Take 5, 6, 7, 8, 9, 10. The number of possible twists and bends and configurations goes up exponentially.
Mm.
Protein folding is a massive technological problem for computer modelers. In fact, we don't necessarily do it directly. What we do is we use rules of thumb. Oh, if you have this type, a whole bunch of this type of amino acid in a row, it'll tend to form a sheet or tend to form a curve or something like that. And we tend to try to image a protein using X-ray crystallography and things like that, because it's really hard to predict what's going to be from the amino acid sequence. Because proteins can form in fact, my protein, the one in my dissertation, green fluorescent protein, was amazing because it folded by itself.
Wow.
Most proteins need help. Most proteins we have, after the amino acid string is manufactured, these molecules, these proteins called chaperones, they grab onto it to prevent it from folding. And they bring it to a molecule called chaperonin, and the amino acid strand goes into the chaperonin. It's almost shaped like a hollow watermelon, and it closes up, and it folds the protein and spits out the folded protein on the other end.
And
Protein folding is a massive problem.
Yeah, I was
And
about to
if
say,
left to themselves, they're not going to fold correctly usually.
so the idea that blind forces created this seems just utterly preposterous at this point.
Yeah.
Okay,
It is. It maybe one or two proteins will be fine, but most of them would not be. They need help folding.
and they need to be a very specific shape in order to be functional.
Yeah, most proteins have there's an okay, we've talked about the genome. That's
Right.
all the genes. There's
Yeah.
other omes. There's one thing called the interactome. It's all the interactions of all the proteins in the cell.
Mhm.
Most proteins interact with multiple other proteins. So like maybe a protein is shaped like this. Well, there's a protein that comes in and it grabs onto this part and deactivates it. There's another protein that comes along and cuts this thing off. There's another protein that opens this up. There's another protein that might add energy. And these proteins, they fit together like locks and keys.
Mhm.
But you have multiple locks and multiple keys at each stage. If you change the folding, you probably destroy the whole system.
Wow.
The complexity is through the roof.
And that's just one aspect of life.
That's just one aspect of many, yeah.
Wow. Yeah, I think my students are going to be excited to see this part.
Cool.
In general, what do you see as the most important issue in scientific Christian apologetics right now? For you, what's the biggest issue that you think most people who aren't Christians or even within the community struggle with?
Oh, it's hard to say. It's probably pornography and sexual deviancy
Mm,
because that hardens people's hearts. It's
okay.
probably wow.
If I
I
could
mean,
get back, it was
it's not
specifically,
necessarily evolution.
yeah.
It's secularism.
Mm,
Evolution led to secularism or contributed to it, fine, but it's from top to bottom in all aspects of society, completely ignoring God and rejecting God as a guiding principle for everything.
how can scientific apologetics for Christians help to address this issue? Because that's the area we're studying.
Okay, good. That's a good, better way to ask the question, I guess, or at least
Yeah.
maybe I didn't answer quite well
Yeah.
the first time.
Yeah.
Works like this.
I'll go back and edit if you want. That's fine.
Okay. No, no, no. I don't mind stumbling. I don't mind being a human being.
Okay.
Um, if we can demonstrate that evolutionary science is fallacious,
Hmm.
if we can show mathematically there's no way for these things to occur, if we can show that naturalism cannot explain origins, then we have an opportunity to introduce a replacement.
Hmm.
I wrote another article. I called it Slaying Yesterday's Dragons.
Hmm.
One of my favorite things I've written. It was basically, yeah, look, we've killed naturalism. We've killed Darwinian evolution. It's dead.
Hmm.
But that doesn't mean that people are going to start believing in creation,
Right.
because what's going to come up is Gaia.
Yeah.
The universe is actually alive. Or emergent complexity. Complex things they just come right out of the fabric of the universe. That's why complex structures arise. Or, as you mentioned earlier, aliens did it.
Yeah.
There are always replacements, but they're metaphysical. They're not scientific. They're philosophical. But that's what we have to get people. We have to get to the point where we can have a philosophical discussion, because this is philosophy and humans are philosophical creatures.
Yeah.
And philosophy influences everything we do and everything we think.
You're making me think about what G.K. Chesterton said. He said, if people stop believing in God, it's not that they believe in nothing, it's that they'll believe anything.
Yeah.
Yeah. Your specialty of genetics and the question of the problem of evil and God's goodness, they call theodicy. From a scientific perspective, does genetics in some way address this issue or help to explain why there's suffering in the world?
Uh, okay, two aspects of that. First of all, I have no issue whatsoever thinking that God created genetic systems perfectly.
Mm-hmm.
So God is a good God. He's a beneficent God. He's a loving God, and he created genetics as a blessing and an amazing thing. And then when we add sin to the world, after Adam rebelled against God, God essentially said, okay, now everything's going to operate according to the second law of thermodynamics, and it's going to fall apart over time. And genetic diseases start happening. Genetic problems start happening. There are people that have deletions of DNA, and it prevents them from having children with certain other people. or they have a deletion that causes a disease, or a broken, a single letter change that gives sickle cell anemia or hereditary blindness or hemophilia. These are, our system breaking down over time. But God did not create these diseases.
Mm.
The diseases happened after that perfect creation was formed.
So it's kind of like Augustine's idea of evil as a privation. It's a lack of what's supposed to be there. So that
Interesting.
genetic diseases would start with a good thing, the genome, and then be a privation, something missing or corrupted within the genome. So that in that case, evil wouldn't be a thing that God did to the genome. It would be a lack of good in the genome because of God's withdrawal of his care over that
Yeah.
genome. Okay.
If
Uh,
Adam hadn't sinned,
yeah.
bad things never would have happened. So the way I think of it, God was providentially upholding the universe so nothing bad would have ever happened. So Adam could not have tripped on a root and fallen off a cliff and died. Never would have happened.
Yeah.
Adam's DNA never would have mutated in such a way to give him cancer.
Mm-hmm.
But upon the fall, God let go that providential watch care. I mean, it's still watching over it in a certain way, still powering the universe, but he's allowing things to proceed according to what we call, you know, entropy.
So what's his ultimate goal in all this?
Well, part of his judgment is long-term suffering.
Right.
Part of the suffering we have now is a reflection on the future eternal suffering in one sense, but also through suffering, we often turn to God. I mean, how rotten would I be if my life was perfect? If I had all the money in the world and I had good health and I had a good education and, I wouldn't have no need for God at all.
Yeah.
I'd have no care for God. I probably wouldn't even be considering it. But suffering has often been led to people to cry out to God and find that God.
Mm.
So one way to a sinful person's heart is by bashing them down.
Kind of the no atheists in foxholes type idea that
Yeah.
that when you're at the bottom, that you realize that you're not in charge of your life and you turn to the one who created everything.
Yeah.
Okay. Any last words for budding apologists? Any words of encouragement or ideas or motivations or directions you might want to give?
For the future scholars of the world, I can tell you that there are still some low-hanging fruit. There are things that the theologians and the philosophers have not tackled because they're not thinking necessarily biblically. And there are very few biblical thinking scholars out there. Therefore, there's a lot of ground that is still unplowed. So go out there and start plowing, start figuring things out, start writing and researching and studying, because there's a lot of work for us still to do. I'm just in our office last week. We're like, wait, no one's ever written about this before. And we're looking through the scholarly literature. It's like, no one's ever written this. And then we're thinking, wait, what are we doing wrong? The idea that we came up with a brand new thing no one's ever thought of is probably not true. And yet, because we couldn't find it, maybe we just discovered something and we just wrote an article about this amazing new thing.
Yeah.
And you can do that too.
Well, excellent. Well, thank you so much, Rob, for being on the show. I
Very
think the
welcome.
people watching it and the students will get so much out of this, and we wish God's great blessings on you, your family, and your ministry. Thank you for
Thank
being
you.
on.
You're welcome.